Thank you, Professor. Now I would like to introduce the Program Development Officer at Mission Lucidity. Mission Lucidity is a research and development hub whose aim is to tackle neurodegenerative diseases. With us, Dr. Jenny Seccarini on the topic, how neurotechnologies will revolutionize research, diagnosis, and treatment of brain diseases.
Good evening, everyone. I'm really happy to be here, and thank you for attending this event. So after Sebastian, I hope that I'm gonna be able to keep the level quite also after you, Gaetano and team. But let's start with Mission Lucidity. So Mission Lucidity is actually a partnership between Caio 11. Sorry. First, I wanted to start with the video. Sorry.
Consciousness is what we are made of. Thoughts, actions, feelings, memories, emotions. And then it slowly fades away. Dementia takes away the essence of who we are. Every 3 seconds, someone new experiences the gravity of this. During our life, we laugh, we love, we live. As scientists, it is our mission to see that we all can do that. And not just for a couple of decades, but for an entire lifetime. It is our mission to stop neurodegeneration. How? By looking closer. Because the next big revolution in neuroscience is actually extremely small. Smaller than the size of a single cell. Thanks to nanotechnological innovations, we will be able to zoom in on questions that science has been trying to answer for several years. It enables us to detect neurodegenerative disorders in the early stages. Combined with clinical and biomedical innovations, we will be able to understand dementia and begin to develop adequate treatments. This is a goal that can only be accomplished by combining our strengths. Therefore, we will share our research and tools openly with all those interested in cooperation. The solution is within reach. We promise to do what it takes to decode dementia. Let us launch this mission. Mission Lucidity. Most likely, the journey of our lifetime. Definitely, a promise you'll never forget. Mission Lucidity.
So I hope that you enjoyed our intro video. So here you hear about our keywords in Mission Lucidity, like nanotechnology, single cells, collaboration, dementia. So is what I wanted to tell you before. So it's a multidisciplinary collaboration between the VIB, the Caio 11, UZ 11, and IMEC. So these are the 4 key partners, founding partners of Mission Lucidity that has been actually launched in 2018. So as you know, we are physically close by, so it's easy to work together, and we try to connect all those experts that really they want to tackle neurodegeneration thanks to their expertise and infrastructures. So with the backing of visionary donors and by leveraging the collaboration, so some research programs together, we wanted to create transformative tools to accelerate both medical discovery and towards a future without degeneration. And how do we do so? We want to combine best-in-class patterns in nanotechnology, neuroengineering, biomedical research into an open innovation ecosystem. So 11 is one of the best places to do that. But also by using industry standard mass manufacturing techniques, such as that the care, all the scientific community can benefit from our tools. And when we talk about industry standard mass manufacturing, we are talking, consider like the standard, the high-level standard of IMEC. So IMEC is one of the key partners for that we rely for nanotechnology. Here I show you the key APIs that belong to our scientific committee. So you can already recognize Sebastian from NERF, Bart de Stroper, that I'm sure you all know regarding Alzheimer research. Patrick Verstreken is the director of the VIB Sense, director of the Center Brain Disease this research. We have Peter Janssens from KU Leuven, Mathieu van de Boelkel, that is a psychiater, and actually is the chair of the scientific committee of Mission Lucidity. We have Rick van den Bergen, actually is the PI, which brings all the expertise from the memory clinic, is really the closest PI and clinicians for all recruiting all our patients in our projects. Ingeborg Stalmans from ophthalmology, and from IMEC we have Dries Braken, that is R&D manager, life science department. But many more other PIs are from IMEC and KU Leuven, and our institution are working together with some projects that actually belong to Mission Lucidity. So, which is our goal? Our goal is to bring, to create these tools within the next 10 years from now on. So these tools to bring it to the clinic and to have a last impacting on prevention, diagnosis and treatment for all those people that are suffering from brain disorders, but especially we start with neurodegeneration. And also the value of Mission Lucidity for our partners is represented by four characteristics. So actually we are connecting, as I said already, the scientists, clinicians and engineers, but we are also supported by visionary individuals that they really strongly believe in our projects, in our mission, so they support our research. We try to build and accelerate powerful, successful research collaboration, so supporting grants and putting together people with the same vision. So we try to combine the expertise and see what we can do together. And also visibility through high-level international collaboration. This is an example. So we are also part of QRND. So it's catalyzing a united response in Europe for neurodegeneration. So together with the DSNI in Germany, Paris Brain Institute, and UK DRI, we form this alliance to tackle neurodegeneration. So this is our research portfolio. So it's we are trying to make a tool-driven revolution, ranging from screening, modeling, stratification, early diagnosis and treatment. So we are trying really to tackle every phase of the disease, starting from the screening, for example, with the personalized genetic risk profile project. For modeling, we have more an advanced chip-based organoids. Maybe I don't know if you are familiar with organoids, but is now something that it's really hot topic, I would say. But one of the closest project to chip-based organoids is one of the most mature projects in Mission Lucidity is Brain on a Chip. I will tell you a bit more in the next slides. And then we have detecting disease retinal imaging, because it's based on the eye as one of window into the brain. It's one of the most accessible organ. And also for early diagnosis and for intervention, I will talk briefly about a new method that we are working on to preserve cognition, so to stop the memory decline in Alzheimer patients. So we'll focus today mainly on 3 projects, not going to detail, is impossible. So we start with Brain on a Chip, and is mainly focused on Parkinson's disease, but can be applied to any brain disorders. So actually, why focusing on Parkinson's disease? We have more than 10 million affected by this disease. I will not go to in much in detail after also the presentation of Gaetano talking about DBS and PD. But we have now L-DOPA treatment has been discovered like 50 years ago, and actually is one of the main most treatment used nowadays, and but is the effectivity of the treatment goes down over the time, and actually progresses causing dementia. So it's not one still the best treatment to stop and treat Parkinson. There are different key problems in Parkinson's disease if you want to study it. So we have limited access to the human brain. This is different between from oncology. Oncology, you have the brain tissue from the patient post-mortem, also from Alzheimer, you can study a bit more, but from like we have it done once we have the brain tissue, you study already it's already reflecting the end stage of the disease. So it's already too late if you want to do something, because the 10 years before that the motor symptoms are appearing, so before the diagnosis is made, there are already other ongoing clinical symptoms that cannot be detected in an objective way. So the diagnosis happening is in a late disease stage, and also also by using preclinical models nowadays, so there is a lack of patient-specific experimental models. So are, okay, still in vivo, so not in vitro, not ex vivo, but still not human. And what we wanted to do with Brain on a Chip, so we are starting with the technology based on the induced pluripotent stem cells, but we are not taking these iPS cells alone, because normally are growing in isolation, but we wanted to create, based on this methodology, we wanted to create neuronal circuits that underlie the disease symptoms and study and manipulate them, record them, activate those neural circuits. So when I talk about the specific circuitry that is affected in Parkinson's disease, I'm referring to 3 specific regions in the brain. So we have the midbrain, namely substantia nigra, where actually the dopaminergic is released to also the striatum. And when the dopamine release and dopamine neurons are already lost at the end, once you have the diagnosis, you have half of these neurons are already missing, already lost. And actually we have these 3 connections. So you have the cortical excitatory neurons that are mainly glutamatergic, connected to dopaminergic neurons, and as I said, the striatal medium spiny neurons. What we wanted to do in Brain on a Chip actually is to recreate the brain network that is affecting PD. So what I just show you, starting from the human stem cells. So at VAB together, in the team of Patrick Verstreken together with Stanford University, there is this collaboration going on already since 2-3 years. There are established protocols to do so, but the key aspect of this project is actually that we are able to differentiate, to create these different 3 types of neurons, and to use the CMOS-MEA chip technology of IMEC. So we're gonna put and let grow these neurons on a chip, and this chip actually can allow us to analyze single cells. So is you can already appreciate, maybe I can use, yeah, of course, the pointer. You can already appreciate the size, you see the size of the electrodes, so are much smaller than our neuronal bodies here. So this can access and analyze, we can really study the single neurons, also thanks to the 16,000, more than 60,000 electrodes on this CMOS-MEA chip. So we can really say that 1 electrode corresponds to 1 neuron. So we really look at a maximum also signal-to-noise ratio to access to these neurons. What we can do with this system, actually, with this CMOS-MEA chip, we can manipulate and measure these human circuits. Here you see a funny smiley, but it's just to show you that you can activate specific neurons, that electrodes that you want. You wanted to record, you can record neuron activity, we can stimulate neurons up to an hour and a half, and also electroporate neurons. I don't put all the exemption. Yeah, this is an example now that our results that actually the in vitro generated neurons communicate on 1 chip. So here we are posit the different neurons, and after 14 days you can appreciate already here there are these different striatal, these different type of neurons are already starting making really synaptic connections. So this is something that, yeah, it's quite unique for this. But also now what we are doing is actually to record, as I said, now is an example up to 5 minutes. We record the voltage, so all this firing rate for the neuronal subtype over time, and we can, the idea, actually we have this recording for all the 3 different neurons, and actually the idea is to extract those features in human controls and to compare them and to see where are the difference in Parkinson. So what we are doing now is to do we are phenotyping Parkinson patients and to see also how different genetic mutation are affecting these circuits, and to see this is the next slides here actually is how we're gonna use this brain on a chip is actually to map Parkinson-specific electrophysiology signature to define patient subgroups, because there are more than 18 types of DNA mutation in different PD subgroups, and to facilitate drug development. So actually to go and develop and let them actually study which is the effect of a new drug on really the neuronal cells. And okay, as I said before, once we have this tool, this platform, the idea is really to share it at the community level. And another project, as I said before, is to using the eye as a window into the brain to do early diagnosis, to perform early diagnosis for Alzheimer's disease. So now we move from Parkinson application to Alzheimer. I have here a brief actually slide summarizing the main goal of the ADMIRE, is the actual abbreviation of our project. So the dimension is just a tsunami. We have now more than 50 million affected from Alzheimer's disease nowadays, but this number will go up tremendously in the next 30 years. So why? Because actually we are still lacking of a quicker, easier, cheaper, less invasive biomarker and a diagnostic tool. So what we are trying to develop is a new biomarker that actually you can screen an elderly population or even people with mild cognitive impairment, so it's the early symptomatic stage of Alzheimer's disease. You can do it in a cost-effective way, because actually we are just using a specific sensor, a specific camera that you can attach to a standard camera system that is used in ophthalmology. So we are always getting tested to see our vision. It's just a 3 centimeters cubic camera that you can, it's actually, yeah, you cannot see her, but yeah, something is missing the picture. And we are, so this technology is based on hyperspectral imaging. So the information is acquired across a wide range of wavelength of light. So here in the, when we measure this hyperspectral imaging, we are referring to an hyperspectral signature of the tissue. So in every time that we have, for example, in Alzheimer's, a myeloid deposition in the brain, this, let's say, CNS Alzheimer's signature is also reflected in the retina. So there are a lot of reviewing of papers showing now this recent also approach to study the retina as a new way also to look at Alzheimer's, but many years before that, maybe this amyloid deposition is already in the brain, you can detect it also with PET or so that is more expensive compared than an hyperspectral imaging procedure. So this is one of an example. Actually, is our first clinical proof of concept using this hyperspectral technique. So we have scanned 17 patients with Alzheimer's compared to 22 elderly controls, and actually by using the hyperspectral signaling only, we could distinguish normal controls from AD patient. And we found actually that if you combine different retinal signatures, such for example OCT image, the sensitivity, so the discrimination between AD and controls increased. So actually here you will see the ROC curve is not extremely perfect in terms of sensitivity and specificity. We are just reached now 75%, but now what we are doing is using actually to further optimize the technical setup using more advanced hyperspectral sensor and new AI related approaches. So it's something that we are working on it to increase the sensitivity. As a last, I would like to conclude with the project based on intervention. So first we heard about DBS. Now we are talking about a method that actually is based on temporal interfering electrical stimulation. So where actually we are using an high and low frequency, and is also we are able to reach the deep regions that are affected, for example, in Alzheimer's, like, for example, hippocampus. So now we are running preclinical experiments in monkeys, and actually what we have found that monkeys that are performing, for example, memory task during electrical simulation with specific intensity, like we are talking about 3 milliampere, or specific frequency, for example, 40 hertz, are behaving much better than during the no ECS conditions. So actually the memory is improving during the ECS. It's still in a preclinical phase. So what we are doing next this year, hopefully also next year, to start new clinical trials in epileptic patients and also in Alzheimer patients. So all these projects are really close in collaboration with IMEC, as I said before. I would like to conclude with thank our founding agency, supporter, all our people and academic institutions that are really working closely with the PIs in Mission Lucidity. And also thanks to you to be here. If you have any question, please, you can also contact me at the booth. We are there.